This presentation on GWASpoly is sponsored by the Tools for Genomics-Assisted Breeding and Polyploids Training Workshop and funded by NIFA, the National Institute of Food and Agriculture within the USDA Specialty Crop Research Initiative, and was held on January 15, 2021.
Okay, so now we're pivoting from talking about linkage mapping to association testing. And this, the name of the package that we had developed to do this a couple years ago is GWASpoly. And I just want to acknowledge the first author on this paper, Umes Roshara, was the co-author of the software with me, and went on to work in the CIMMYT breeding program, and as part of the Gobi project, I believe. So we, as part of the SCRI, over the years we've gotten requests for, oh, wouldn't it be nice if we could add this feature and this feature, and now we finally have some funding as part of the SCRI to implement some of these improvements, and so we're going to come back to that at the end. But right now I'm just going to tell you about basically what the software, as it was created a couple years ago, can do. And a lot of this talk is just going to focus on wrapping our heads around how is GWAS different than linkage mapping, because it seems like it's not that different, but there are just so many complexities that come up when you start to try and do regressions in these kinds of populations. So here is a table that kind of starts us thinking about that. So both GWAS and linkage mapping are techniques for QTL discovery, right? Sometimes people will say QTL mapping and they mean that to mean not GWAS. I personally don't like that terminology because both in my mind are methods for detecting QTLs. I tend to use linkage mapping or GWAS to be the way I differentiate those two different settings. So in terms of what's going into the regression model, the difference here is shown on the first line. The predictor variable is different. In the GWAS, the predictor variable is the actual allele dosage for your marker. So 0 up to whatever the ploidy that of your species. And GWASpoly does allow for higher order ploidies, unlike the diaqtl. So there's really the marker data feed into the model in a kind of unmodified form, if you want to think about it that way, at least for the additive model. So for the additive model, this is the predictor variable for GWAS, and for linkage mapping, it's the dosage of the founder haplotypes. It's not the dosage of the marker alleles. And that's the critical step that the linkage analysis allows you to take the marker data and transform it to draw inference about those founder haplotypes. So that's the difference from an additive model. And when you talk about dominance models, again, there's a difference between, say, a dosage. It's not of the allele itself, but it's a dosage of some function of the allele, of the marker allele in the GWAS, and it's the dosage of a diplotype or a higher order combination of founder haplotypes, but the same principle. And then as far as what the type of population that you can apply it to, the GWAS in principle can be applied to any population, and that's the appeal of it. I have this asterisk here just as a reminder to say that in theory anything, but in practice that flexibility can have consequences, and the more you understand those consequences, you may come back and say, well, maybe I shouldn't have created my population that way. But we'll see if we can attain that kind of wisdom by the end of this short talk. For the linkage mapping, you're talking about a pedigreed population where you can track all of your offspring back to a set of founders. And in the case of tetraploids, at least at the moment, the software we have only allows one meiosis to separate the founders from the offspring, but it's certainly, in principle, possible to imagine kind of chaining those kinds of analyses on one another. And I think we'll see that technology is created in the next couple of years as people try to look at multi-generational data sets. So the big picture idea going into this is that GWAS seems straightforward to do, and it is, but then that flexibility comes with some limitations compared to linkage mapping. So when does GWAS work well? There are three conditions that I've shown here. First, it works best when you're talking about QTL that are approximately biallelic. Two, the minor allele frequency for the QTL should not be too low. And three, you have lots of unbiased markers. Now let me elaborate on those 3 points a little bit here. So the first one, that your QTL is approximately biallelic. This admittedly is a bit of a circular argument, but if we accept that, for the most part, the kinds of marker data sets that people are using are biallelic SNPs, although you can get beyond that. But if you are using biallelic SNPs, then you're relying on them to have LD with your QTL, and there's only so much LD you can get between a biallelic entity and a multiallelic entity. They just don't line up. So it's not a question of frequencies even, you just have the wrong number of levels of your factor to get a high correlation. But if you have approximately biallelic QTLs, then you're more likely to find some markers that have approximately an allele frequency and an LD that matches that QTL. Second point, this reinforces some of what Chris said at the beginning about, okay, what is the determinant of statistical power in a QTL analysis? The heritability due to the QTL, or the proportion of variance, is certainly one important thing. The LD between the marker and the QTL and the population size. And if you look at what affects the heritability of the QTL, one of the main drivers is the allele frequency at the QTL. So the higher the higher the frequency of the minor allele, the more, all else being equal, meaning if you have the same effect size and you have the same amount of measurement error, you're going to get more variance explained by the QTL. So that's the reason to try. That's why it works better when you don't have QTL with very rare alleles. And then lastly, lots of unbiased markers. You need ideally you would have about one marker per haplotype in your data set per effective chromosome segment, meaning it's sort of statistically independent, right? And what that number is depends on how you've constructed your panel. But if you have, for example, a very diverse panel with land races or wild species, you're easily talking in the hundreds of thousands of number for kind of markers that you would need. So linkage mapping, there's almost no benefit practically to going above 10,000. Yes, I mean, you can see benefits, but it's diminishing returns, right? You can do a very respectable linkage mapping with 10,000 markers, and you could get lucky with GWAS and make it work with 10,000 markers, but it might not work. You might need 100,000 or a million, and that's part of the bit of the gambit that you take when you try and do a GWAS. Okay, this next slide here is a little bit of a technical point, but I think most people are aware of the concept that you typically do some filtering of your markers based on their minor allele frequency before you run the analysis. Why do you do that? This is just to explain the rationale for that a bit better. We've talked about how you need high LD between the marker and the QTL for the marker to pick up the QTL when you do your scan. And a QTL, a marker with a low minor allele frequency is really useful for detecting a QTL with a low allele frequency, but you may not have power to detect that QTL because it doesn't explain enough of the variance. And therefore, keeping all these markers around that have a frequency below the value that you can even detect as QTL is wasteful from the perspective of the computational burden. And also means that you're just inflating the number of tests that you're conducting, and you have to then keep ratcheting up your threshold to control the false positive rate. So that in a nutshell is why it's wise to filter your data set. Now, exactly what the threshold should be, that will depend upon your population size. In general, the larger the population, the more you can tolerate having the better ability you have to detect a rare QTL. So you may want to start to keep markers that have a lower frequency. What about inferring the causal gene? Can we do it, right? I mean, if we didn't, I wouldn't say that's the only reason to do QTL analysis, but certainly besides realizing the benefits for selection, we mostly want to try and understand what it actually is, are the genes underlying these quantitative traits. And this can be quite complicated in a GWAS. In linkage mapping, it's far more straightforward. The reason being that the the LD between your markers and your QTL in a linkage mapping context is only due to linkage. That's why it's called linkage mapping. As a result, as you march steadily closer to your QTL, the marker significance, the lot score, is going to get higher and higher. And this allows you to more or less know where the QTL is. Now, again, measurement error tends to complicate that simplistic description, but given a large enough population with enough markers, there's really no limit to how precise you can narrow the interval for a linkage mapping context. And that's why it's the preferred method when it comes to actually trying to do map-based cloning when you don't know a lot about the underlying molecular biology. In a GWAS, other factors besides linkage or proximity between the marker and the QTL are contributing to LD, such as allele frequency differences, differences in the evolutionary history, or this might sometimes just call it like haplotype structure. As a result, the most significant markers in your analysis may not be the ones closest to the QTL, and then therefore to go from the significant marker to developing hypotheses about candidate genes, you have to have a much better understanding of the molecular biology and a good reference genome with good annotation to try and make further progress. And those conditions are certainly met in many scenarios, but it's important to be aware of those differences. And just to illustrate for you how this plays out in a real example, here is that 3x3 potato half-diel, which I can analyze in two different ways. I can analyze it using a GWAS methodology. So the black circles are using the GWASpoly software to analyze it. The trait in this case is maturity. And the way we represent significance is minus log of a p-value. And then we can analyze the same data set using the linkage mapping software, and that's the blue, which is on the right axis. And if you look at this kind of at this level, this is looking at the entire chromosome 5. You have the impression that, oh, the GWAS is giving me a much better resolution. I mean, the region of the chromosome where you see some upward gesture of the circles looks to be a lot more narrow in the black than it is in the blue, where you see kind of this broader peak. And so people, including myself, sometimes make statements like, oh, well, you get better resolution with GWAS because you get to harness all the historical recombination. And that's true, but it's deceptive because when you really get down to it, in my experience, it's like getting to like the 5-yard line, if I'm going to use an American football analogy, which probably some people will cringe at. You get to the 5-yard line, and then you have a hard time figuring out where the end zone is. Here's the zooming in at a 1.5 megabase interval, and the black circles, you can see the most significant markers in the GWAS. They're about 100 kb away from where the gene is. We happen to know the causal gene in this example, and the peak in the lot score is the plateau is right exactly at the causal gene. And if you look at that dashed red line, you'll see there are markers that are actually in the gene, and they have almost no significance. So again, this is because of these differences in allele frequency and haplotype specificity that confound the attempt to use physical distance as a reliable indicator in a GWAS. All right, population structure, kind of a related concept to things we've talked about already, but I'm going to present it in a more statistical light here. What does it mean? Everyone's heard this when you hear about GWAS. Basically, if you are combining groups that have different evolutionary histories together and you want to do this regression, the problem potentially is that any genetic difference between those groups can be confounded with the genetic differences for that trait, if that trait is also different between those groups. So for example, maybe these two groups have diverged and there's been some selection on this trait of interest. And so you want to try and capture that, but they were selected. There were other traits under selection, or maybe there was some genetic drift because the populations are small. And all those other markers affected by those other evolutionary forces are going to be confounded with what you're looking for, unless you find a way of controlling for it. And that's what people refer to as controlling for population structure when you do a GWAS. And the most efficient way of doing that is to include a random polygenic effect in the model, to a kinship matrix that you estimate from the markers. So this kinship model was originally developed by some plant breeders back in 2006, plant geneticists who, brought a new perspective to what up to that point had primarily been the province of human geneticists. And this ushered in the era of mixed model analyses for GWAS. Now, before that and afterwards, you still have this other idea that you can use fixed effects based on principal component analysis or a model-based clustering algorithm. And of course, the most popular one is the software that is called Structure in capital letters. These analyses produce covariates that you can use, and those work, but they generally don't work as well as K. So K would always be your first approach. And then if you need to add P or Q in addition to K, there are situations where that can lead to slight improvements in power when your trait is strongly correlated with population structure. But I can tell you that in practice, I never do that because we're talking about very small improvements. And from a practical perspective, it's good for writing a paper, but it's not good for much else in my experience. Okay, what about the detection threshold? So we've already spent quite a bit of time on this idea. I mentioned already that the nice thing about linkage mapping is that basically you can do a simulation and the results are relevant for whatever species and traits you may be looking at. And that's not true here because there's essentially an infinite number of ways that you could construct a population for a GWAS. So the most foolproof method is to do a permutation test, which is computationally demanding. But there is a function to do that within the GWASpoly software. And as an approximation to that, you can use other methods, the most common being a Bonferroni type of correction. I think most of you probably are familiar with this. Bonferroni means take your desired significance level divided by the number of tests that you want to do. In this case, since we're talking about minus log p, that would be your threshold. And the Bonferroni method has the property that it is a sufficient but not necessary approach to controlling the false positive rate, meaning that it's overly conservative. And the reason is that the different markers that you're testing are not independent, and therefore you may be able to use a less stringent threshold and still do a good job of controlling your false positives. And you don't want to make the threshold any higher than it has to be because that just means you're losing statistical power. So one of the ideas is that in the Bonferroni formula, replace the total number of markers with an effective number of markers. And this is actually an idea that goes back to human geneticists working in GWAS 20 years ago. And there are a number of proposals about how do you calculate an effective number of markers. One of the objectives of the SCRI project was to implement one of these methods because it wasn't part of our original software. And actually I just managed to do that last week. So that's one new improvement for GWASpoly compared to the original software. You can now opt to use an effective number of markers compared to the total number. And I'll show you in the vignette what benefit that has. So if we look at the options within the software, this function has the obvious name set threshold, and the default now is this mif for effective markers. Besides the Bonferroni method, which at this point I don't see a reason anyone would want to use that, but it's just there for backwards compatibility. The permutation, the other method included in the software is called FDR, which stands for false discovery rate. It's the proportion of significant markers that are false positives. And when you first hear it, it sounds like, wait, isn't that the same thing as a false positive rate? It's not. The false positive rate is actually a probability of making any false positive when you control it on an experiment-wide basis. That's the technical definition, whereas this is a proportion of the markers that were called significant that are in fact truly null or false positives. The practical point is that the difference between the FDR and the FPR is very small for GWAS, and in my experience, not so much worth worrying about. The FDR methodology was developed originally in the context of microarray for expression studies, so EQTL or those kinds of analyses, in which case you expect a large number of genes typically to be upregulated or downregulated when you do those kinds of experiments. And therefore there is a big difference between them two, but in a GWAS where maybe 1% of your markers may be significant. That would be your expectation. That's how many would be truly significant. At best, there's really not much difference between these two different approaches. Now, with the exception of the permutation test, which is a robust method that even when you have departures from certain assumptions works because you actually construct the null distribution directly. The other methods, meaning the Bonferroni type methods or the FDR, they assume that the p-values you're calculating are actually p-values, which sounds nonsensical, but the reality is that the statistical tests that you can do in the GWAS, even though the software tells you it's a p-value, it actually may not be a true p-value because true p-values have to follow certain theoretical distributions. In particular, under the null hypothesis, the p-values have to be uniformly distributed. What that means is if I were to imagine a graph of the distribution of p-values, and I look at, say, the box, if I bin it between, say, 0 and 0.1, so I look at how many p-values are between 0 and 0.1, and how many markers have a p-value between 0.9 and 1, the number of markers in those two bins actually should be about the same under the null hypothesis, and that's what we mean by uniform distribution. If you do a naive regression without correcting for population structure, you will not find that that's the case. In fact, you will see that the distribution of p-values is inflated relative to the expectation. And fortunately, when you do correct for structure in a good way, it more or less takes care of this problem. But this is one of the signature diagnostics that you should do when you run a GWAS, is just to make sure that your p-values are not inflated, and therefore you can apply these types of methods for controlling your false positive rate. And the function to do this in the software is called QQplot. QQ stands for quantile-quantile. And many of you probably have encountered the idea of using a quantile-quantile plot when you talk about checking the normality of the residuals. In that case, the expected distribution is a normal distribution, but it's not normal in this case. So you can't actually, most of the functions for QQ plots, like in basic stats packages, will use a normal distribution. So you can't use them here. You have to use one that's specifically written to check it against the uniform distribution. So this is what the output, the figure generated by the software looks like. The dashed line is the one-to-one line. So under the null hypothesis, all of your markers, all of your p-values should fall on that line. And this is showing you what was actually observed when we analyzed tuber shape with that half dial L in potato. The red is the K model, and you can see how it hugs that dashed line pretty well until you get to pretty high pretty high scores, minus log p-values. And then you get this strong deviation, which is what you expect, that you want for most of your markers to be null, not significant, and then you get a couple that are far off that line. Now look at what happens in the blue when you don't use the structure correction. You just do an ordinary linear regression like you would use in a typical in a linkage mapping analysis. You can see that you get this, it's like floating above the line, it's inflated. And that's one of the signatures that means you haven't properly done your structure correction. The other main visual output from the software is the Manhattan plot. That's the name of the function. And here you can see the implications of not correcting for structure in this example. So the left is the kinship model, the right is the uncorrected regression model, and we get this nice peak on chromosome 10. That's the QTL that we saw already in the previous seminar. But if you draw, say, a horizontal line at a threshold and you use this effective number of markers, in the corrected model, you don't find any other QTL besides the one on 10. In the uncorrected model or naive model, you get these peaks that they just come right above the threshold, and you would conclude that, oh, maybe there are some other minor QTL, but those are actually artifacts of your analysis. So there are a couple different genetic models that can be implemented in the GWASpoly software. The ones I'm going to focus on here are the additive, simplex dominant, and the general model. So the additive model, we've heard several times already in different contexts. It means that your phenotype is proportional to allele dosage. And in this case, the dosage is of the SNP allele or the marker allele. Here's an example of a trait in potato that shows a highly additive response with respect to the dosage of one particular marker. This is potato maturity and the CDF gene on chromosome 5. In both years of our trials, we see this proportionate reduction in maturity as we increase the number of copies of the SNP allele. And what about simplex dominant? So we talked about this already. This is the other kind of dominance, not the one that's implemented in diaqTL, but it's more similar to the kind of complete dominance that we know from our introductory genetics classes. And there's two non-equivalent dominant models for every given marker, depending upon whether you choose the reference allele as dominant or the alternate allele as dominant. So actually the software, when you tell it to test for simplex dominance, it's actually doing two genome scans, one with the reference allele dominant and one with the alternate allele dominant. And then it combines those results for the Manhattan plot, but it shows the different models separately when you look at the QQ plot. This is the result for tuber shape, again, using the kinship corrected model. And in this case, the QQ plot, all three of those models, they hug that dashed line, so we're not worried about inflation. And then all of them seem to produce some significant markers that deviate. And if you look over here in the Manhattan plot, we can see actually that there's both the additive and the dominance models are producing significant results that have a similar score, a similar minus log p value. And when we first developed the software, we had this naive idea, I would say, that, well, you could look at which model produced the most significant markers, and then you could use that to interpret what was the actual genetic gene action at the actual QTL. Like, does the model that produced the most significant result at a marker, does it really tell you something about the behavior of the QTL? And it does if the QTL is biallelic. But when the QTL is not biallelic, it can be quite hard to interpret the results. And I think that's what's going on in this particular example. This is where the linkage mapping clearly has an advantage because it can accommodate multi-allelic QTLs. And this, if you recall, was the result from the linkage mapping in that 3x3 diallel, where we saw there was one haplotype on this parent that seemed to be different than the others. Then maybe there's a second allele for alleles haplotypes 2 and 4 in the first parent. Looking at these haplotype effects, I would guess there's at least 3 different QTL alleles present at this locus. And I think that's why we don't get as clear of an understanding of the gene action just by looking at the single SNP results in the Manhattan plot. The last kind of model is a general model. And as the name implies, it actually includes all the other models. It has the flexibility to assign a different genotypic value for each different dosage value. So the dose 1, dose 2, dose 3, and 4 are all arbitrary relative to the dose 0. So the number of degrees of freedom is equal to the ploidy when you try and run this model. And that additional degrees of freedom means that you have slightly less statistical power than if you just used one of the simpler models, which have a single degree of freedom. So there's a trade-off between the flexibility and the power. And I guess the key point here is that when you start to run multiple models, just be aware that whatever significance level you specified for each individual model now technically doesn't apply because you're running more and more tests. So you need to either accept that you're going to have a slightly higher false positive rate than what you specified for each model, or you need to reduce that significance level for each one of your models if you still want to have the overall control to be at your target. In practice, we find that the additive and the simplex dominance often are good enough to find what we're looking for. Okay, so last slide here about some of the theory and the background. We are planning a number of improvements to the software, as I mentioned already, as part of this SCRI project. The first one is to add some more simulation tools to the package itself. I mean, we can do all these things on our own, but I think to enable other researchers who don't maybe have that expertise to do it themselves, we're going to build some functions so that you can, say, try and determine what statistical power do you think you have for a QTL of this heritability for your particular data set. and that may help you with designing or interpreting your results. The other major area of improvement is to try and expand the functionality for GBS markers. I mean, the software does work for GBS markers. Many people have used it, but it was really not designed for that. We mostly have worked with potato SNP array data in my group. And so we want to, for one, allow a VCF file as an input file type. We want to account for the uncertainty in the allele dosage calls that come from different read depths in a polyploid. And lastly, we want to potentially allow for multi-SNP haplotypes, multi-allelic markers, and the Polyrad software already has the ability to export VCFs with that kind of information. So we're looking to use some of those example case studies to try and build out this functionality for the software. And lastly, we heard a number of requests of people who want to try and analyze polyploid and diploid data sets together, populations together, or perhaps tetraploid and hexaploid. But anyways, right now you can only analyze one ploidy at a time. You can scale the markers yourself, and there are some pros and cons to doing that, but we're going to try and make it a little bit more user-friendly to do that kind of an analysis. All right, now I'm up to the point of doing the vignette. So the package is on GitHub now. For the last couple of years, it was just on our lab website, and I migrated it to the GitHub site a few months ago, so that we could start to make improvements again. And there's the vignette, of course, and the reference manual. So let's move on to what do the input files look like. So the genotype input file, the first three columns are your map, marker, chrome, position. And what you're seeing here is an example of a reference genome position. It can work with you can use a centimorgan, or I should say, you can use a genetic map, but I think it doesn't allow decimal places. So if you have centimorgans, you may need to just shuffle your decimal place over, like for example, reported in millimorgans or something, something other than centimorgans, just so it's a whole number. But I know when people worked in barley, we didn't have a reference genome, and we were using consensus maps from a couple different mapping populations to do some GWAS. So it can work just fine like that too. The subsequent columns are the marker data, and the formats are shown here. You can list it, the actual nucleotide calls, and that's the ACGT format. You can use the AB concept, like you would have from an infinium array or axiom array. And then you can also then just represent the dosage of whatever allele you choose. It does, the software doesn't really care. Whatever the number is here, it's going to call that the alternate allele. And that number has to vary between 0 and whatever the ploidy is. And it does allow for non-integer values. So that has the advantage that if you do some imputation method with some other software, it may give you fractional values because that's a better predictor than a whole number. You can use the posterior mean estimates of dosage instead of the posterior modes. And for example, Polyrad has the ability to output VCF files with posterior means. So those are some of the advantages of using the numeric format. The phenotype input file looks just like the input file for the diaqtl. The first column is your clone name, then your traits, and then any covariates after that. And in diaqtl, we made the decision that when you read the data, you tell it, okay, is this a factor or is it a numeric covariate? In GWASpoly, you specify that using a function called setParams instead of the reading the data. So just a different decision about where you specify it, but either way, you have to tell the software how to interpret those other columns. And then this table just outlines what the functions are in the basic workflow, which is at the top of the vignette. I'm not going to read all these right now, but we'll just start at the top here and read in the data. So the vignette data set is not the 3x3 DLL. It's an older data set from a type of diversity panel in potato, but it really was more heavily based on elite germplasm and cultivated germplasm. You can get the names of the files. They were called table S1 and S2. Then you say read GWASpoly. You tell it what ploidy you're working with, what's the format of your marker data, how many traits are in the data set, and then it knows based on that, okay, how many columns of covariates are there. And it will give you some output saying, here's how many markers we found, number of clones. These are the fixed effects that we detected, excuse me, and these are all the traits. The next step is to set the kinship matrix, and in hindsight, I would have made this not a separate step. It would have just been automatic when you load the data. That's, for example, what we did in diaqtl. You don't have to explicitly tell it to create the kinship matrices. But when we wrote the paper in 2016, we were very interested in the question of, do different types of kinship matrices make any difference? And basically the answer was no. But we'd already sort of written the software based on that idea. So you can give it a different kinship matrix estimated with a different software package, or you can just let it do it internally, in which case it just does the kind of computation that you use in genomic prediction, where you take the allele dosage matrix and multiply it by its transpose. So that's the set k function. Then if you want to change any of the parameters of the analysis, you have to use the set params function. And this includes adding covariates. So in this case, if we want to use these fixed effects to look at what we'd call the P plus K model to control population structure using a type of principal component, loading on a principal component, we would have to do that here. If you want to change the minimum minor allele frequency, which is the default is 0.05, you would change that here. If you want to change the maximum genotype frequency, which is not something that you hear a lot about, but it turns out that once you start to look at simplex dominance models or duplex dominance models, you can find that you just don't have enough instances of the two different classes to make the test worthwhile. And so you can reduce the number of tests and improve your thresholds by modifying that. The default is 95%. No one genotype class can account for 95% or over 95% of the instances. And the last thing you can modify is whether you want to use what's known as the P3D approximation. Now, the P3D approximation means that the variances are only estimated once, and then it uses actually a generalized least squares test for all of the markers, which is faster, but it does have slightly less power. So if you want to disable that, you have to do that here. Okay, so the function to actually run the scan, the genome scan, is just called GWASpoly. You give it the data the output from the read function, and you now tell it, okay, which models do you want to test? In this case, we'll say additive and simplex dominant. Those are the two that I usually use. And then of the 13 traits, let's just test these two of them. Now, in diaqtl, in the previous seminar, you can only pick one model or one trait at a time. You have to basically write your own for loop to try different models and traits. And it's just a decision about how complex to make the output or whatever. In this case, at the time we wrote the software, we decided to put the for loop internally, so you can tell it to test a bunch of different models and traits, and it just has a for loop inside that does all this, and it gives you some output as you go along, that it's analyzing this trait and it's testing under this model. And as I mentioned already, when you tell it simplex dominance, it actually is testing two different models, whether the reference or alternate allele is the dominant allele. And because I didn't pass that params variable, it's just going to use the default, which the default is the K only model. If I want to run the P plus K model, I have to add into the function this params variable that I generated when I called set param. So they aren't talking to themselves directly. You have to actually give this as an argument to the function. So let's see how the K versus the P plus K performed as it relates to p-value inflation. The function, as we said, is called QQplot to generate this type of an output. And QQplot returns a variable of the GG in the grammar of ggplot2, which means that you can just add more layers on top of it like you normally would. So in this case, I'm just going to add the layer that put a title on the top of this, and I say this is now my K-model result. And you can see that all three of the different models, they pretty much have no inflation. So the K-model seems to be working quite well. And this is the QQ plot result with the P plus K model. If I toggle back and forth, I can't really see any difference between these. So there's really no reason to add complexity to the model. We're just going to proceed using the K-only model. Look at the Manhattan plot. It creates a paneled plot using the facet wrap functionality of ggplot2 for the different traits, and the different models are shown in the same graph using the different symbols. In this case, this is a different data set than the one we looked at before. In the 3x3 dialog, remember that there was like the simplex dominant model and the additive model both had markers that were similar in their scores at that chromosome 10 QTL. This is actually an older SNP array where we don't have as good of a marker set, and you can see we're missing some things. We're not picking up anything with the dominance model. Of course, the population is different too, so that could be part of the explanation. But I think primarily it's a reflection of some of the ascertainment bias of this original SNP array. All right, so after you've looked at your results that way, then you can maybe want to get a table output. And so say, okay, actually, which markers are above my threshold? Well, first you have to set the threshold. That command is called set threshold. You tell it what method to use, and the older Bonferroni, which uses all of the tests that you do, that's the first thing that we'll look at. So if we want to see what is the threshold that's computed under this method, we actually haven't written a function that tells that to you, or it doesn't even display it. We could do that, but if you want to see it, it's just called threshold. So you can just use the @ sign. In R, there are two different types of classes. They're called S3 and S4. The S3 classes, you use the $sign. GWASpoly uses an S4 class, so you have to use the @ sign to access all the elements. And if you're ever in doubt about something, when you get something from a function and you wonder, what is this thing? I always just type str, and I give the name of that variable, which is called structure, and then you'll see all the guts of that function, and you'll know exactly, okay, if you want to dig inside of it, what is there? Of course, that's considered bad practice from a software design perspective, but we're too pragmatic to worry about that, I think, in this community. So what are the thresholds? From the Bonferroni method, you can see they're slightly different for the additive versus the simplex dominance models. And this gets at what I was alluding to earlier. When you convert from an additive to a dominance model, you can actually suddenly now be above the threshold that you've set for maximum genotype frequency. So it's actually dropping out some of the markers in the test, and that's why these thresholds are lower than under the additive model. If we use this new effective number of markers approach, the thresholds are lower uniformly compared to the Bonferroni method, and again, that's the benefit of using the M effective compared to the original Bonferroni. But it's a small difference. These are not huge differences. Once you've set the threshold, when you call getQTL, now it knows what markers to export as being significant, and it produces this table, which has information about the markers, including what is the ref versus alternate allele, which if you give it to the ACG, if you give the marker data in the ACGT format, it actually doesn't specify which is that in itself doesn't convey what's the reference and the alternate, and the function just uses an alphabetical sorting to do that. When you use the AB format, A will always be reference and B will always be alternate. And when you use the numeric, it will always call 0 as reference and 1 as the alternate. But it does give you, and it kind of wraps around here, but you can see that the last column of this table is the effect. Now the effect is the regression coefficient, and the interpretation depends upon which model you're talking about. So in the additive model, the effect is the change in the phenotype as you substitute a C for a T. So you change the dosage by 1, and this is the effect on the average effect on the phenotype, basically. So in this case, it means that the tuber shape elongated by zero 0.3. And if it's a negative number, the phenotype is decreased by that amount. So that's what was present in the original software. Now, as we already had a question about where is the R squared value, and that is by far the most common request that we've gotten about where we need that because publications want to see it and so on and so forth. The problem is that it's a complicated, it seems like a simple question, but it's a complicated answer because R squared doesn't have a kind of universally accepted meaning when you talk about mixed models. And even though I know there are other software packages you can go to which will show that to you, the reality is that you have to look under the hood to find out what are they actually reporting. The advice I've typically given people is, well, after you've gone through this process and identified what are your significant QTL, build a multiple linear regression model using a stepwise regression method. And there are any number of software packages, including an R, that will do that for you, and you can get your partial R squared values from that kind of analysis. So I just haven't wanted to sort of bring that additional step within the fold of the GWASpoly software. But I've reconsidered lately, and I think we're going to try and build a function called fitQTL, which will attempt to do this. I haven't fully decided on what methodology to use yet, but hopefully in the next year, we'll try to provide some additional functionality to give it a list of significant markers, and it will at least report the proportion of variance for those different markers and the polygenic effect. So hopefully that will help people interpret some of these results. So to sum it up, I think I like to think of GWAS as a quick and dirty method for gene discovery. You know, it's interesting how there are different fads or trends, let's say, trends in genetic analysis, right? There was a time when GWAS really became, this would be like in the mid 2000s, early 2010, it really became the workhorse of a lot of crop breeders and geneticists. And I think that it's still very valuable when linkage mapping is not an option, like especially if you're, say, more of a pre-breeder, you're working with diverse germplasm, you're not going to create a bunch of populations. It still is a very effective method to get some idea about the genetics of your traits, but just realize that going the last 5 yards from the QTL to the causal gene can be a challenge. You typically need some additional information, understanding of the molecular biology to do that, followed by some type of knockout experiments or other transformation-based experiments to nail it. When you have the capacity to do linkage mapping, and for breeders, you are constantly generating populations that are well suited to that, I think that it's more foolproof, and it's, for me at least, easier to convert those results into something that I can take action on in terms of haplotype selection. So I think I'll stop there and be happy to take any more questions if we have them.
This presentation has been sponsored by the Tools for Genomics Assisted Breeding and Polyploids Research Project. For more information, you can email us at toolsforpolyploids@tamu.edu or visit our website at www.polyploids.org. This project is funded by NIFA within the USDA Specialty Crop Research Initiative funding program.